Welcome to Animal Biotechnology. Today, we are exploring a fascinating and life-saving topic, the production of donor organs. Imagine a world where no one ever dies waiting for an organ transplant. That is the incredible promise of xenotransplantation. And in this lecture, we are going to understand exactly how it works from both a biological and mathematical perspective. Let us start with a simple analogy to understand why this field exists. Imagine you have a classic car that desperately needs a new engine to keep running, but the original manufacturer went out of business decades ago. If you try installing a completely different brand of truck engine, the car's internal computer might immediately recognize it as foreign and shut the whole system down. This frustrating scenario is exactly what happens in human medicine. When a patient's organ fails and there are no human donors available, scientists look to animal organs to save lives. However, the human immune system acts like that strict car computer, instantly attacking the foreign tissue. This brings us to xenotransplantation, which is the scientific term for the production of donor organs from animals for human use. You might naturally wonder, out of all the animals in the world, which one is best suited for this critical task? The answer is pigs. Pigs are chosen as the ideal biological factories for these organs because their internal organs are remarkably similar in size and overall function to human organs. Additionally, pigs grow very quickly and can be bred in large numbers in sterile, highly controlled environments. This ensures a steady, safe, and scalable supply of organs to meet the massive global demand. But there is a major biological hurdle we must overcome before this can work. If a standard, unmodified pig heart is transplanted into a human patient, a devastating process called hyperacute rejection occurs within mere minutes. Why does this happen so quickly? It is because human antibodies are naturally programmed to immediately recognize and attack a specific sugar molecule found on the surface of pig cells. This molecule is called alpha-Gal. To make animal organs safe and viable for humans, biotechnologists have to find a way to hide or completely remove these foreign markers so the immune system does not trigger an attack. To solve this severe rejection problem, animal biotechnology steps in as the ultimate genetic mechanic. Scientists use highly advanced gene editing tools, most notably CRISPR-Cas9, to precisely disable or knock out the specific genes responsible for producing those foreign alpha-Gal markers. But they do not stop there. Furthermore, they often insert actual human genes into the pig's DNA. By doing this, the pig's organs begin to express human proteins. This brilliantly tricks the patient's body into recognizing the transplanted organ as its own native tissue, effectively bypassing the immune system's natural defense mechanisms and preventing rejection. Now that we understand the biological concept of gene editing, let us look at how we mathematically model the survival of these transplanted organs over time. The survival probability follows a classic exponential decay function, which perfectly describes how viability drops under immune stress. In this formula, S of t is the probability of the organ surviving and functioning at a specific time t. S0 is the initial survival probability at the exact moment of transplant, which is typically 1.0 or 100%. The variable r is the rejection rate constant. This is crucial as it measures how aggressively the immune system attacks. A higher R means a stronger, faster attack. Finally, T is the time elapsed since the transplant, usually measured in days. Let us see this formula in action with a concrete example. Suppose a hospital tests a standard, unmodified pig heart. It faces a severe immune attack, giving it a high rejection rate constant of 0.693 per day. We want to find the survival probability after 10 days. We plug our values into the exponential decay model. Multiplying the rate by 10 days gives an exponent of -6.93. Calculating this exponential function gives a final answer of approximately 0.00098. Physically, this means the unmodified heart has less than a 0.1% chance of surviving just 10 days. Now, let us compare that to a genetically modified gal-safe pig heart, where the rejection-causing genes are removed. This lowers the rejection rate constant drastically to just 0.02 per day. Using the exact same formula for 10 days, our exponent becomes -0.2. Because this exponent is much closer to zero, the exponential function returns a much higher value. The final answer is approximately 0.8187. This means the modified organ has an 81.87% chance of survival. This massive mathematical difference proves why genetic modification is absolutely required. While knocking out one single gene is a great start, making a pig organ truly safe for long-term human transplant is much more complex. Scientists often need to knock out multiple genes simultaneously to completely evade the immune system. For instance, they target not just the alpha-gal gene, but also other rejection-causing genes like the CMAH and B4GALNT2 genes. Editing multiple genes at once in a single pig embryo is a massive technical and statistical challenge. Every additional gene edit lowers the overall chance of creating a perfect embryo, which brings us to our next mathematical model regarding probability. To understand the success rate of these complex multi-gene edits, we use a fundamental probability formula derived from statistics. Here, P sub t represents the overall total probability of creating a perfect triple knockout embryo, meaning all 3 target genes are successfully removed. The variable p represents the probability of successfully knocking out any single target gene during the CRISPR process. We raise p to the 3rd power because there are exactly 3 distinct genes being targeted for removal simultaneously. If the success rate p increases, the total probability, p sub t, increases exponentially, which is why improving CRISPR efficiency is so critical for biotechnologists. Let us calculate this for a real laboratory scenario. Suppose the probability of successfully knocking out any single target gene using CRISPR is 40%, or 0.4. We need to find the probability that a single injected embryo will successfully have all 3 genes knocked out at the same time. We take our single gene success rate of 0.4 and raise it to the 3rd power. The final answer is 0.064. This means there is only a 6.4% chance of success for any given embryo, highlighting the sheer scale of work required. Now that we have used this formula to find our success rate, let us understand where it comes from mathematically. In genetic engineering, the editing of each individual gene is considered an independent statistical event. According to the basic probability rule for independent events, the total probability of all events occurring together is simply the product of their individual probabilities. So we multiply the success rates of the 1st, 2nd, and 3rd genes together, shown as P1 times P2 times P3. Because the success rate is exactly the same for all 3 genes in our specific model, we can substitute the variable p for each individual rate.
Multiplying p by itself 3 times logically simplifies the expression to p raised to the third power, giving us our final working formula.
Let us visualize this entire journey. On screen is a flowchart of the xenotransplantation process. In step 1, you can see CRISPR scissors cutting the DNA of a pig cell to remove the alpha-gal gene. In step 2, this edited nucleus is transferred into an empty egg cell. Step 3 shows this blastocyst developing into a fully grown transgenic pig. Finally, in step 4, the heart from this genetically modified pig is transplanted into a human patient, where the immune system accepts it. This diagram beautifully summarizes how genetic engineering bridges the gap between animal donors and human patients. We have covered a lot of ground today. Let us review the most important ideas. First, xenotransplantation uses genetically modified animal organs, primarily from pigs, to solve the global organ shortage crisis. Second, tools like CRISPR are essential to knock out genes that cause hyperacute rejection. Finally, as we proved with our mathematical models, these genetic modifications drastically lower the rejection rate, turning a near-zero survival chance into a highly viable, life-saving treatment. You now understand both the biology and the math behind the production of donor organs.